This video demonstrates a procedure to conduct differential imaging of two Raman resonances associated with biological structures with coherent Raman scattering signals. First, three synchronized short pulse lasers are overlapped in time and space. The overlapped pulses are then sent to a standard inverted microscope, which allows for the generation of coherent Raman signals from microscopic objects. Signals are converted into images using commercial software in order to compare different Raman signals that are produced. Results indicate that the difference between images obtained from doubly and singly resonant Raman signals provide enhanced sensitivity for weak Raman signals.
Hi, I'm Tyler Weeks from the Center for Biophotonics at the University of California, Davis.
I'm Thomas User from the Center for Biophotonics and the Department of Internal Medicine at the University of California, Davis.
Today we will show you a procedure for using doubly-resonant Coherent Anti-stokes Raman scattering for producing chemically specific images of cells.
We use this procedure to detect signals from molecules that are too weak to be detected directly by Coherent Anti-stokes Raman spectroscopy alone.
So let's get started.
In order to generate CARS and DR CARS signals simultaneously, three tunable and synchronized short pulse laser sources are required. To obtain these synchronized pulses, start with a single short pulsed laser with an output power of 10 watts, a fixed wavelength of 1064 nanometers, a fixed pulse length of 7 picoseconds, and a fixed repetition rate of 76 megahertz. Using a series of half-wave plates and polarizing beam splitter cubes, the beam is split into three parts. A half-wave plate combined with a polarizing beam splitter cube allows adjustment of the amount of power in each component of the beam without changing its direction. Typically, two beams are adjusted to approximately 4.5 watts each, and the third beam contains the remaining 1 watt. The two high-power beams are then directed into two independent optical parametric oscillators, or OPOs. OPOs use difference frequency generation to convert a high-energy photon into two lower-energy photons with different wavelengths. By controlling the temperature of the crystal used to achieve this effect, the wavelengths of the resulting photons can be controlled to within 0.1 nanometers. By frequency doubling these signals, a laser with a fixed wavelength of 1064 nanometers can now be transformed into a laser beam that can be tuned anywhere between 780 nanometers and 910 nanometers. By pumping two separate OPOs with the same 1064 nanometer source, two independently tunable laser sources are obtained that are automatically synchronized to our original pump laser. The third beam from the 1064 nanometer pump laser is directed around the OPOs by a combination of dielectric mirrors so that all three beams can be recombined later. In order to efficiently produce coherent Raman signal photons, the recombined pulses must be overlapped both temporally and spatially. Because OPOs contain ring cavities allowing each laser pulse to pass through the crystal multiple times, the beams sent through the OPOs travel an extra distance, resulting in considerable delay relative to the original pump beam. This distance must be compensated for with the third beam by introducing extra mirrors that force this beam to travel the same distance as the other two before they are recombined using dichroic mirrors. In order to provide fine adjustment of the length of the path, each beam is sent through adjustable delay stages which allow for adjustment of the temporal overlap of the different laser pulses. Another set of half-wave plates and polarizing beam splitter cubes are added to each beam to allow adjustment of the laser power of each beam independently. Dichroic mirrors are used to first combine the beams from the two OPOs and then the 1064 nanometer beam. Care must be taken to ensure that the beams are precisely overlapped in space. This can be verified by comparing the overlap of the beams within a few centimeters of the dichroic mirror with those approximately 1 meter from the dichroic mirror. Because exposure to high average power laser beams can damage the sample, the three combined beams are sent through an electric optic modulator functioning as a pulse picker, which allows adjustment of the repetition rate of the pulses that arrive at the sample and therefore the average power. The combined laser beams are then coupled into an inverted microscope with an objective lens with high numerical aperture, or NA. The tight focusing, generated by the high NA objective lens, allows for the most efficient generation of coherent Raman signals on microscopic length scales. The objective lens of the microscope is mounted on an XYZ piezo stage, which allows images to be obtained by raster scanning the beams across the sample, similar to commercial beam scanning confocal microscopes. Now let's see how to generate CARS and DR-FWM signals. The interaction of 3 short-pulsed laser beams at the sample results in the generation of several 4-wave mixing signals, such as CARS from the various combinations of 2 lasers, as well as 3-color CARS and DR CARS signals from the combination of all 3 lasers. When signals are relatively close in wavelength, it can be difficult to separate them for analysis. For this reason, signals are passed into an imaging spectrometer that also serves as a monochromator or bandpass filter to spatially separate signals at different wavelengths. An electronically actuated flip mirror within the spectrometer in the lowered position sends the signal to a back-illuminated deep depletion charge-coupled device or CCD camera, which provides spectroscopic information across the entire signal range and allows us to identify and optimize the various coherent Raman signals. To select the signal to image, simply rotate the grating within the spectrometer using the vendor-supplied control and data acquisition software to center the peak of interest on the CCD camera. Then change the position of the flip mirror to redirect this signal to a second exit port, to which a single photon counting avalanche photodiode or APD is attached. Raster scan the objective lens and then use the signals recorded on the APD to generate an image by displaying the photon count rate for each pixel with the data acquisition software. Repeat this imaging procedure for each desired coherent Raman signal so that signals can be compared during post-processing. Now let's see how to properly prepare a sample. In order to obtain clear, reproducible images, some care must be taken in preparing the samples. Samples are typically prepared on approximately 150 micron thick glass cover slips. These cover slips are thin enough to allow for high-resolution imaging with the high NA water or oil immersion objective lenses that are used in these experiments. To demonstrate the preparation of a typical sample, we prepare approximately 20 micron thick sections of mouse muscle tissue that are fixed to a microscope slide. A 20 microliter droplet of 5 molar deuterated glucose solution is added to the sample. The deuterated glucose solution provides a unique and strong Raman background signature. A glass cover slip is placed on top of the sample and fixed in place with nail polish. For cells in culture, glass bottom culture dishes can be used that allow for imaging without the need to detach the cells from their cell culture growth dishes. Now let's see how to determine the proper Raman peaks for DR CARS. In order to properly take advantage of the doubly resonant enhancement effect, the Raman spectra of both Raman resonant substances must be known. Typically, these are 2,845 inverse centimeters associated with lipids and 2,121 inverse centimeters for the CD stretch mode associated with the deuterated glucose. Coherent Raman scattering is achieved when the frequency difference between 2 lasers matches the frequency of a molecular vibration. By tuning one OPO to 817 nanometers, it will probe the 2,845 inverse centimeter CH mode when combined with the 1,064 nanometer laser beam, and by tuning the other OPO to 868 nanometers, it will probe the 2,121 inverse centimeter peak when combined with the 1,064 nanometer beam. In spectroscopic mode, the CARS microscope allows us to observe 3 coherent Raman signals of interest: a CARS signal probing the CH stretching vibration, a CARS signal probing the CD stretch mode, and a DR CARS signal probing both. In the diagram shown here, dashed arrows indicate photons from the laser and the OPOs, and solid arrows indicate the resulting signal. The solid horizontal lines indicate the energy of the Raman vibration and give a visual representation that, in DR CARS, mixing the same 3 input photons simultaneously probes 2 different Raman vibrations. The signal strength for each peak will need to be optimized by tuning in fine steps around these peak locations. Here we select each peak and take images of C. elegans worms in a solution of deuterated glucose. Extracting additional information based on these 3 individually obtained images requires some fairly simple image processing.
First, the images must be normalized. A practical method for normalization relies on the fact that lipids are hydrophobic. This means that in regions of high-density lipids, the CD resonance from the glucose should contribute very little to the doubly resonant signal. Conversely, in regions of pure glucose solution, the CH resonance from the lipids should also contribute very little.
With this in mind, normalize the DR CARS image and the CARS image obtained on the CD resonance of the deuterated glucose solution to a region well outside the C. elegans worm that should exhibit no CH resonance. Next, identify a region within the worm in the CH resonant CARS image that is rich in lipids and normalize it to the corresponding region within the normalized DR CARS image. For this method to work properly, assume that deep within this region no deuterated glucose is present. This is a safe assumption since the lipids are hydrophobic and will not mix with the solution. Now, by subtracting the normalized CH resonant CARS image from the normalized DR CARS image, just the amplified CD resonant signal remains. Similarly, by subtracting the normalized CD resonant CARS image from the normalized DR FWM image, just the amplified CH resonant signal remains. Shown here is the Raman spectrum for a modified oleic acid that includes an alkyne modification. The strong CH resonances at 2,845 inverse centimeters and the alkyne resonance at 2,121 inverse centimeters are both well isolated from the fingerprint region, which is the region of densely packed peaks, making them ideal markers for coherent Raman imaging. This is a typical spectrum of coherent Raman signals generated when 3 short-pulse lasers are overlapped within the sample. The arrows point to the processes responsible for each signal as represented by energy diagrams. These are typical results from imaging C. elegans worms using DR CARS and CARS. The top row used the 3 signals just shown to image a worm in a solution of deuterated glucose. In the second row, the images were appropriately normalized, and in the third row, the difference images were produced by subtracting each of the CARS images from the DR CARS image.
We have just shown you how to perform Raman difference imaging based on doubly resonant coherent anti-stokes Raman microscopy.
When doing this procedure, remember, it's important to make sure the beams are overlapped and synchronized properly. Also, when switching between different coherent signals, make sure not to bump or move the sample, as this will affect analysis. So that's it. Thanks for watching and good luck with your experiments.